Alright. Okay.
Hi everyone, good morning. So I'll be continuing with chapter 9 with protein kinase C. So unlike what Aksana discussed last time, PKA, this goes with an independent um, phosphorylation from CAM. So protein kinase C is a serine threonine kinase that is involved in many cellular processes. So among these are tumor formation, host defense, embryologic development, pain reception, uric growth, and long-term memory. So how it all started, why did researchers got interested with PKC, is that they found this croton oil, which is this poisonous viscous liquid found in from the seeds of croton plant. So this was introduced in the Western world by the Dutch from China. So they initially used this as uh, an exfoliant, skin irritant, but then they found that it is tumorigenic and skin irritant also. And then the active principles they found in the croton oil are the 4-bol esters that activate the protein kinase C.
When you say it's poisonous, do you mean that it causes tumorigenic and skin irritant, or does it does it actually kill some organisms or whatever?
Oh, I'm not sure about how drastically it can.
Okay. I just wondered.
They have this.
That's really interesting.
Yeah.
Okay.
These effects.
And when year was that? Like, do you know around when this all came?
Like the
this
research?
Oil? Yeah, this research.
The research started around 1980s, they said, but the oil has been around for like maybe
so we didn't know about
ago.
protein kinase C until 1980.
Yeah.
Wow.
Because
that's pretty recent history.
Yeah.
I mean, it's only 40,
and I actually
40
checked
years.
eBay. It's still around. Like the croton oil is still around, but it's used
And what's
for
it?
external use only. They exfoliate. They use it as an exfoliant. I don't know, but yeah, they should.
That's
know.
interesting.
Yeah.
Okay.
So yeah, it's still around. And then so 4-bol esters were found in that croton oil, and then it actually competes with the physiologic activator of PKC, which is dimethyl. So this is the AG. This is just a small molecule, but then compared to the PMA or the 4-bol esters, which has this long hydrophobic groups, which makes them easily diffuse through the plasma membrane and do their actions. So these are the red ones are the important residues that make it resemble the action of the AG. And then, so the protein kinase C family is comprised of 9 genes coding for 11 distinct enzymes. So they are grouped into a family according to structural similarity. So these are the C3 and C4 domains are the conserved regions. So C3 is the ATP binding site, and the C4 section is where the substrate actually binds. And then they are further grouped into subfamilies according to their activators. So the conventional subfamily, which is CPKC, are activated by all 3 DHE phospholipids such as phosphatidylserine and calcium. So this is the conventional. So they have this. So this is the catalytic region, which is conserved for all 3 types, and they differ in this part, which is the regulatory domain. So the conventional subfamily have all the 3 binding sites for DAG, 4-bol esters, and calcium, C1 and C2. The novel isoforms would have only their activators as DAG and phosphatidylserine, not calcium. So instead of the calcium C2 binding here, they have an extended C1 domain. And then the typical isoforms would only have phosphatidylserine as their activator. So they don't have the other the C1 and the C2 domains. Instead, they have these other nuclear associated proteins, which are their activators.
So they call it conventional, novel, and
this is
atypical.
Conventional, novel, atypical. So
Okay.
the conventional alpha, beta, and gamma, and novel is delta epsilon.
Oh, got it. Okay.
Gotcha. So yeah, so I had to consult the
Yeah, me too. Yeah,
to actually review the Greek alphabet.
yeah. I hear
So,
you. I can
yeah.
say it, but I don't know what all the letters look like.
Yeah, so those are the letters.
Yeah.
So for me to talk about it, I need to review
Yes.
the Greek alphabet.
Yeah, I hear
Yeah, so those are the subfamilies. And then looking into the structural domain, so we said the C1 and the C2 domain are the regulatory features. So this is the part of the protein, C1 and the C2. And then so the C1 attaches to the plasma membrane through the TEG or formol ester, which is an activator. And then the C2 binds the calcium, both of them. And then there's a hinge region which connects the C2 to the catalytic domain, which is C3 and C4. So the C3 and C4 are the portions that phosphorylate cerebral threonine residues, which are near arginine residues. So this is the consensus sequence, which is the book said is typically found in many proteins. So by looking at it, we can see that maybe it's not very specific because this is very commonly found in many proteins.
Yeah.
So we will see later what renders specificity for this phosphorylation.
So for this typical or conventional.
Yeah, this is a conventional.
Yeah, conventional. Sorry, you need both calcium and
diacylglycerol.
diacylglycerol or formol ester.
Yeah. So there's another illustration in the book that
Okay.
shows how it is activated
Yeah.
actually. And then so those are the C1, C2, 3. And then there's another region, the turn and C-terminal hydrophobic motif, which actually binds to the only known upstream kinase that regulates PKC, which is PDK1 phosphor. I have the
Yeah, it's okay.
I have
Yeah,
it here.
protein something
Yeah.
kind.
So the phosphorylation of PKC by PDK1 must happen before the enzyme becomes fully competent to phosphorylate its substrates. So so it controls the conformation of this area of the
What
PKC.
activates PDK1?
I haven't read about it, but
Okay, yeah, I just wondered.
yeah, there must be something else
right.
upstream.
Mm-hmm.
And then I found this interesting component also of the structure is that it has a pseudo substrate, which is the psi, denoted by the psi symbol here in the N terminal, which serves as a lock mechanism. So when the enzyme PKC is not active, it actually folds around the hinge region. So remember, you have the hinge region connecting the C2 and then the C3. So what makes the folding possible is the pseudo substrate. So they named this as pseudo substrate because it resembles the consensus sequence earlier, this one, except that it has alanine instead of serine. So it can bind to the catalytic domain, but phosphorylation does not happen. So it's just there to hide the catalytic domain when it is not active. So yeah, without stimulus, binds the catalytic domain to fold the protein. And then another is the RAC binding site, which is RAC stands for receptor of activated kinase, C kinases, which is at the C terminus here. And then the pseudo RAC motif, which also serves as a lock mechanism when the enzyme is not active. So if we remember from Vivek's talk, he talked a lot about IP3, right? So and how it induces or stimulates calcium release. So from there, the other product produced during IP3 synthesis is DAG. So PLC actually is the enzyme that produces both IP3 and DAG. The acylglycerol is the activator of PKC, the conventional or the novel lectures. So what happens to DAG is that it diffuses around the plasma membranes. It is hydrophobic. And then the calcium stores released by IP3 would bind to the C2 region of PKC, whereas the DAG, this blue thing, will anchor the C1 domain towards the plasma membrane. So initially, an inactive folded PKC is just hanging around. It's somewhere near the plasma membrane, and it's associated with cytoskeleton. But then when this initial stimulus happens, then they are anchored to the plasma membrane by DAG, and then calcium binds, and then conformational changes happen that releases the lock, the pseudosubstrate, to expose the catalytic domain. And this is where PTK1 acts. So this is the activation. It's called the activation segment. So once it is phosphorylated, then it becomes fully competent to bind and phosphorylate PKC substrates. So yeah, and then this one is the negative regulator, so diacylglycerol kinase. So when it's done with its work, so DAG is phosphorylated by DGK, so that serves as a regulator of the process. So the PKC is then released from the plasma membrane to fold back to its inactive form. So it can be reused.
That's really cool.
Yeah, it's really cool.
So, what recruits it to do? You know what recruits it to the membrane, or just kind of recognizes?
So this is the main thing that they discuss: the release of DAG,
Okay.
the acylglycerol.
So that's what.
That's what it captures.
It's amazing.
Yeah, I find it amazing too.
Yeah, like how does it know?
Yeah, exactly. I mean, everything that we've studied so far is like
It's so
pretty
amazing. Yeah.
Like, how do they know where to go?
How do yeah,
How do
exactly.
you know when it's time to go?
Yeah, right. Yeah. I mean, what is the
Yeah.
That's interesting.
Very
Okay.
interesting. Anyway, so that's cool. And then some sources of activators. So like so we earlier we talked about DEG and IP3, but where do they come from? So these are the lipid sources that becomes the source of diacylglycerol. So PIP2, and then which is acted upon by PLC and PA3 kinase, and then they have distinct isoforms that they do activate. So CPAC is conventional and novel, and then the other PA3 kinase mediated is just novel and atypical, and then somehow I think it gives some specificity in terms of which activation happens, which isoform is activated because of the specific specific lipid sources.
Young has a question.
So in the negative CPKC means the upstream is inhibiting. Okay.
Yeah.
Okay.
Yeah. So that's the only inhibitory factor. And then take a look at also arachidonic acid is also an activator of CPK. So later we will see that they use it in the paper that I will be discussing. So Aksana mentioned during her talk that PKA tissue specificity is rendered by this region, right? So this portion, the RR, makes it tissue specific. But this is not something found in PKC. Instead, they localize into different cells, different subcellular compartments, and they bind to unique protein complexes. So somehow that renders them specific. I mean, instead of having this regulatory. Yeah. So these are just the list of different tissue distributions for the different isoforms. And then they also bind different sets of proteins, which are also what they phosphorylate. So these are their substrates. So depending on what the substrate is and depending on the function, then that will be the prevailing physiologic process that will be acted upon by disease. So there's a lot of it. So that also makes PKC a very important for many processes. So we talked about the activation. Now we turn to what deactivates the process. So we said earlier, the presence of the AG kinase, when the AG is done with its work, will break down the AG2 phosphatidic acid, which will stop the membrane localization of PKC. But then the signaling events do not stop there, because phosphatidic acid itself is involved in DNA synthesis, rough recruitment, and all these processes. So it just stops the localization. But still, these events continue to happen. And then another form of deactivation is, as we said earlier, the 4-ball esters, when they for prolonged activation by 4-ball esters actually tends to inhibit PKC activity, because what happens is they can dephosphorylate the critical residues of the protein itself and degrades the protein eventually. So it somehow balances itself when there's too much 4-ball esters. So that's about PKC.
Are there endogenous 4-ball esters?
No.
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 Okay.
So the only known physiologic activator of PKC is the acetylglycerol.
Oh, okay.
Yeah.
That's the only one. And
Yeah.
with the calcium. Yeah.
Yeah, the calcium.
Yeah. Okay.
Yeah, so the paper I chose talks about riluzole, which is a PKC inhibitor for amyotrophic lateral sclerosis. So I think one of the recurrent themes that we've seen in this class is that many of the drugs that are actually marketed now we see that they actually have more than one mechanisms of action. So this is one example. So the PKC inhibition activity of riluzole was later on just discovered. It was not the original
Right. I teach it as a
of action.
antioxidant, I think.
Yeah. Antioxidant. But it actually touches on that mechanism also.
Oh.
This particular disease.
Okay.
So, so amyotrophic lateral sclerosis is a progressive and fatal neurodegenerative disease of the motor neurons. And so what happens is that the motor neurons are degraded somehow, that they cannot contract, and then it becomes fatal when important muscles such as respiratory muscles becomes affected. So patients die. So what happens here? Well, there are more than one mechanisms of the disease, but the prevailing hypothesis is the cortical hyperexcitability, which is mediated by the glutamate neurotransmitter. So glutamate excitotoxicity is the prevailing mechanism, and it is also used as a diagnostic marker to differentiate it from the other neurodegenerative diseases. So that is how riluzole was first described as an inhibitor of NMDA release. So it acts on the sodium channels to prevent excessive glutamate release in the presynaptic vesicles, presynaptic terminals. So that was its first mechanism of action. But then researchers found that not only this mechanism is important, they also found increased oxidative state has been found in the ALS patients, and very important, I think this is an important paper that connected how PKC activity is involved in ALS. So they found increased activity of the enzyme activity and the level of enzyme in ALS patients. So that's why this paper that I'll be talking about investigated if PKC is actually involved in the action of riluzole also, not just the NMDA release. So we talked earlier about the free fatty acids, which are the sources of the AG. So excessive free fatty acids will activate protein kinase C through the AG, and then one of the substrates it can act on is NADPH oxidase, which eventually leads to oxidative stress through release of free radicals. So this is the main mechanism of the disease that this paper focuses on, and how ribosol actually attenuates neuronal death. So earlier we mentioned about arachidonic acid. So they use that particular compound to induce neuronal death in cortical tissues. So they found that ribosol and GFX, which is a selective PKC inhibitor, both decrease the neuronal death caused by arachidonic acid, and they measured it in terms of LATH release, which is a marker of cell death. So yeah, that's the first. And then after that, they did measure PKC activity in both the membrane fraction of the cortical tissues and in test tube assays. So in both setups, they observed the same trend, that ribosol and GFX inhibits PKC activity.
Can you tell me what the bars are? Because I can't see from here.
So this, the black is blank, and PMA is the formamidostine acetate,
right,
which is the
a formyl ester, right?
Yeah. They use this as activator of PKC system.
And what's on the x-axis? What's that say? What's the measurement?
Okay, membrane PKC activity.
Okay.
The way they measured it is they have this neurogranin substrate, which is a known substrate of PKC.
Okay.
And then they measured radioactivity of the phosphate group that becomes attached.
So it's a radioactive acid.
Yeah.
Okay. So
so they have ATP, and then it had
P32 or P32.
Yeah, P32.
Okay.
Yeah.
So then the formyl ester PMA activated, and then what's the next 3 bars?
Okay, so this is ribosol at 30 micromolars and ribosol at 100 and GFX at 1. So this is the known.
Those are high doses, though.
Yeah.
And what cells are these again? I'm sorry.
These are cortical cultures from rat brain.
They're primary.
Yeah.
Yeah. That's a very high concentration for primary cells.
Okay.
Yeah, you know, right?
Yeah.
Okay, all right. And then what's this one?
This section is test tube assay. So
it's isolated enzyme.
Yeah, isolated. PKC itself, like purified PKC form.
Okay, and then what are the same concentrations?
Same.
So even in the isolated enzyme assay?
Yeah.
Oh,
Yeah.
that's a lot of drug. I'm just saying, like.
So what's the ideal range for primary cell culture? For drug?
Well, for any drug, it's really nanomolar.
Oh,
Yeah.
okay.
Yeah. In cells, micromolar is okay, but 30 is pretty high. Usually 10 is like the highest micromolar.
1, 3, and 10.
1, 3, 10. If it goes above that, it doesn't mean it's not involved. It's just a very high dose. And then in the isolated enzyme, they usually go down to like nanomolar doses. I'm surprised they had to use so much for the isolated enzyme. Yeah.
They did discuss in the paper that these, although I'm not sure how they could measure that, it is a lot less than the physiologic concentration that is reached.
Oh.
Like when the patient actually takes it, it's a lot less than what goes into the blood.
Okay. Okay.
But yeah, they commented on that.
Okay.
But that would be like different, like its whole body, and it's just very small cells and have a direct effect.
Yeah.
And I have a question to that question, like the cortical cells you are talking about. It's only neurons, or they have taken all the cortical
They have
cells?
neurons and glial cells.
Neurons and glial cells, all. So
So they don't try to make pure neuron culture. It's a true cold culture.
Yeah. And then they also did immunoblotting, but they didn't quantify.
Yeah.
So this actually just tells us that, because remember we mentioned that prolonged activation of PKC will actually degrade the enzyme itself. So this is what is seen here after 6 hours of exposure with the purple ester, and then the PKC active inhibitor, positive control, prevented that, and the same for riluzole. So I think they were just trying to say that the action of riluzole and GFX does
Are
not
similar.
happen because of it destroys the enzyme, but
got
it acts somewhere in the enzyme. And then that's what actually they did in the following experiments. Oh, but this one is another
Oh, yeah.
photomammogram. I don't know, but this is the control, the sham wash control, but I think it looks more. The white spots should indicate the cell death. And
Oh,
then-
that's the white spots is where so they're doing LDH release.
Yeah,
Okay.
LDH release.
Okay.
And then, so this is after 48 hours exposure to formyl ester, and then riluzole and GFX.
So less
Okay.
release is less cell death.
Less releases, yeah. They do have that
Yeah,
quantification
so
this time.
2 control and then the PMA. Yes. And then PMA plus riluzole and GFX plus B. So what are the groups? I'm sorry.
Okay, so this is PMA only, formyl
Oh,
ester only,
Yeah.
PMA with riluzole,
Oh yeah.
and PMA with GFX. So they trying to see if riluzole
Yes. Yeah. If both of
can
them.
counteract the PMA-induced death.
So it's not as good as the GFX,
Yeah.
and it's also a higher dose.
Yeah.
So plus 10 plus 30, but it does inhibit. Yeah. Okay. Yeah. The cultures look clumpy.
Yeah. So yeah, they did next try to investigate if actually riluzole binds somewhere in the PKC protein. So PDBU is a PMA analog which they use to check if remember we have the regulatory domain and the catalytic domain. So this section of the experiment they used to check if PKC binds in the regulatory domain because that is where the PMA analogs will bind. So this graph shows us that. So this is the measure of binding of PDBU, and then the decrease in this trend is seen because of the addition of PDBU itself. So at displacement of
The radioactive.
the radioactive.
So they use
the
cold and
cold,
cold,
yeah.
non-radioactive to
Yeah.
compete with the radioactive.
Yeah.
Yeah.
So they saw a decrease in the binding, but it was not seen in riluzole addition. So it means it does not affect that part of
Okay.
the protein binding.
Right.
And then so they next check the catalytic domain. So PKM is actually you can break the PKC into regulatory and the catalytic domain. So the PKM can stand by itself
Oh.
and actually still catalyze enzymatic reaction. So they did that here, similar with what they did earlier. The neurogranin substrate was that they measured, and then they observed also decrease in they observed PKM activity was decreased with riluzole and GFX
Yeah.
addition. So this time they're seeing there's
Just
some
with the
effect.
with the catalytic
catalytic
domain. Yeah, just
domain.
making sure their assay worked.
Yeah, yeah.
Okay.
And then so the other portion of the catalytic domain, the C3 domain, is the ATP binding site. So they check if additions of ATP will actually decrease PKC activity, but it did not happen. So they're saying that riluzole action could not be on the ATP binding site
Hmm.
itself.
Yeah.
So ATP
So it's
can
not on
still-
the regulatory domain.
Not regulatory.
It's not in the ATP binding
Yeah.
site. Okay.
So
it's like
they ended there.
Yeah. Oh, they didn't go on to figure it out, huh?
Yeah, because this is an earlier paper, like
Yeah. Yeah.
but I tried to dock the retinol
Wow. Yeah.
compound itself through the catalytic
Oh,
domain.
cool.
I wish I could show you where it actually binds, but the browser is not working.
That's
But
okay.
these the really high docking scores
Okay.
tells us that it actually binds in this portion of the.
And what portion is
this
that?
is the iota isoform catalytic domain.
Okay.
So
So it
but
binds
yeah, I could
somewhere on the
still not
catalytic
show it,
domain,
yeah.
just not in the ATP
yeah, just
site.
not
Yeah, so it's made what we would call a type 3 inhibitor, right? An allosteric inhibitor.
Yeah.
probably.
yeah.
Yeah,
It's
yeah.
what also they speculated.
Yeah, is that what they
Somewhere
expected in the
there.
paper? Yeah, somewhere.
Yeah.
That's really cool.
Yeah.
That's cool you went further and did that. That's neat. Yeah.
Mm.
What was I gonna say? I don't know. It just left my head. Someone else asked a question, so it just left my head. I don't know. Okay. I don't know.
That will be the last. And so yeah, just to summarize, PKC is a serine kinase that acts independently from CAM and is involved in many cellular processes. And relating it to the disease of interest, ALS, PKC activity leading to oxidative stress is implicated in that particular disease, and that riluzole serves as a neuroprotectant by PKC inhibition.
Yeah.
So
Cool.
yeah.
That's very good. Let me see your references. I just wanted to see when they're from. Okay, yeah. So your paper was from 2000.
This one.
Yeah. I wonder if anything's been done since then. Did
Yeah, I
you?
was also trying to check.
Yeah, you didn't see anything since then. Yeah.
I'm also like fascinated why they still wanted to do it, because it's not like they have a competitor for this particular drug, because
Right.
it's the first drug that has been
Used for
approved
ALS. I know. Yeah.
Yeah.
It's still. Is there another one, Matt? Do you know?
I found that there's another one, but it acts on a different mechanism.
Oh, okay. Because I teach about riluzole, but I can't remember if I teach. I think there's another one. Yeah. ALS is we need drugs for ALS for sure.
Yeah.
Interesting.
Yeah, they say that there's no treatment, they only delay.
Yeah, exactly. Delayed expression.
It's a fatal disease.
What?
It's a fatal disease.
Yeah,
and-
actually, so Rehana and I, Dr. Leek and I, we worked in the same lab at the University of Pittsburgh for a while. We were both research assistant professors, non-tenure track faculty in this lab of Michael Zigman. You probably know his name from reading his papers, maybe. He just passed away of ALS a month or two ago. Yeah. So.
There was one person in my country, like I know him, and he suffered from ALS,
Yeah.
and he died within 5 years.
Yeah, it's well, so it can be, it's either rapidly progressing or slow progressing. So have you ever heard of Stephen Hawking?
Yes.
He had the slow progressing ALS, so obviously he lived a long time with it. And then I had a friend in college who actually Dr. O'Donnell also knows. So weird, like all these connections. She worked with him obviously after he graduated college, but I knew him pretty well in college. He had ALS, and he had probably like a medium progressing form because he lived till his 40s, and he was diagnosed soon after college, not too long after college. So
Like he was diagnosed, that person which I'm
friend?
talking about it, he was diagnosed in 45 or 46 years of age, and he died around 51.
Yeah.
50 years. And it was
Yeah,
rapid.
rapidly.
Like for the last 3 years, he was completely paralyzed. He was his brain was active. He can talk through a gadget, like with eyes direction.
Yeah.
Other than that, his whole body cannot
Right.
move, and then he finally died just because maybe of the respiratory.
Respiration.
Right.
Eventually, it affects all the muscles, not just skeletal
Yeah.
muscles, unfortunately. Yeah, and Dr. Zigmund, who I just mentioned, he was diagnosed 1 year, a little bit more than a year before he died. But he was, he was in his 80s, probably 80s, maybe. So he was elderly, but still, yeah.
Is it genetic? Like, is there any specific reason?
Well, yeah,
Some
there are some thoughts that there are some genetics involved of superoxide dismutase, which is the enzyme that helps reduce oxidative stress in all of us. There's some evidence to suggest that some of these people have a mutation in that gene, so it doesn't handle oxidative stress as much. And maybe I don't know, it doesn't seem to affect only older people though. I think, no. So I don't know, And I don't know what the switch is, like, because my friend was fine all through college, you know what I mean, I knew, and then all of a sudden after college, it's like, boom.
He was fine till 45 years old. He was not diagnosed with anything. And I think what, like, there is an insult or stimulus, which basically
some environment. I'm
initiates
sure it's
that.
a combination of environment and maybe some genetics. I don't know.
Yeah, every neurodegenerative disease is
It's
like
both,
that. Like
right.
it's at some point it starts progressing, and we don't know because it's progressing because it's the initial stage.
Yes.
And everything is done in those initial days, and the symptoms appear way after that. So that's the most difficult thing to.
It's hard.
Diagnose and then treat.
Yeah.
At some point, it becomes untreatable, and you cannot do anything.
I know,
Other than palliative treatment.
Yeah. So I've known 2 people, and the one of the surprising things or concerning things about Dr. Zygmunt is he worked on this Parkinson's disease. And on the floor where he worked on Parkinson's disease, there's another person who has Parkinson's disease, who was also a researcher in Parkinson's disease. Two of his colleagues died from Parkinson's disease, who were also big Parkinson's disease researchers. A lot of them worked with rotenone, paraquat, things like that.
That's really harmful, really dangerous things.
So it's really interesting. Well, I mean, it's, you know what I mean, it's interesting from a scientific point of view. Of course, it's not something you want to happen, but the fact that all these researchers that worked with this disease have gotten something, I mean, ALS is not exactly the same, but something similar, to that.
But it's a good thing if you are dying with Parkinson. At least you are living till 80 years of age.
Yeah. That's true.
Not like other things like ALS is very dangerous.
Right. Yeah. Very interesting. Now, so your paper's from the 2000. It made me think of this thing I just recently saw on one of the social media sites. Someone posted that they're a professor at a university, and a student asked them, "Is it okay if I use a paper from the late 1900s?" And the paper was like 1995. She nearly died. I'm like, yeah, I'm dying now too. Thank you very much. The late 1900s, 1990. How far back can we go? I'm like, oh my, I'm so old. So when you had the 2000 paper, it made me think of that. I'm like, oh, that's almost the late 1900s.
I think this is the first time I'm hearing someone referring 1990s
Late
as 1900s.
I know. Oh my gosh. What? Oh
That's
my gosh.
the 2000,
Yeah, it's just
actually.
like, I'm
Millennium.
so old.
That's millennium.
Anyway.
Yeah, that's very good, Melanie. Thank you.
And I actually have a little surprise.
Oh.
Yeah, just want to thank everyone for
Aww.
Thank you.
I enjoyed learning with all of you.
Oh, good.
And, thank you for making my first semester a lot more bearable.
Aw, yay. Thank you.
Thank you, Melanie.
Well, that's a cute picture. Where were you guys?
That's in Taj Mahal.
We went to
Taj
Taj.
Mahal. Where's
Yeah.
that? On Craig
Doors-
Street?
McKnight.
Doors
Oh,
here.
McKnight. Is there one on Craig Street?
I don't know where we did go.
That's near the Rachel's home.
Yeah, it's Ross Township.
Rose Township.
Ross Township. Is it good at Taj Mahal?
Yes.
Yeah, it was.
Yes,
amazing.
yes, yes.
Yeah. Oh, that's so nice you guys got together. Oh, thank you. Yay.